which are around 100 and billions. And you will also have some supporting cells, the cells that support the structure, the cells that support the framework, which we call as neuroglia. Okay, so neurons and neuroglia. So neurons we almost studied during skeletal muscle. How is a neuron? There is a soma, there is an axon, there is a dendrite. Okay, so we know in a neuron there is a cell body and there is a long axon, and then there is a terminal bulb and there are some dendrites. We know about neurons. So there are almost around 100 billion neurons all over in the brain. And what is in the neuroglia? Neuroglia, they are supporting cells. They are supporting cells. Alright, so what is neuroglia? So we start with neuroglia today. What are neuroglia? So neuroglia are supporting, they support the brain with a lot of functions, and also anatomically and physiologically they support the brain. So there are 4 different types of neuroglia. They are oligodendrocytes, then there is astrocytes, then there is microglia, and there is ependymal cells. So there are 4 different types of cells which are found in the brain, and they support the neurons and support the functions, also give some structural stability, support the framework, and do many functions there. Also a protective nature. Alright, we will discuss it one by one. So there are 4 different types of neuroglia: oligodendrocytes, astrocytes, microglia, and ependymal cells. Right? So we start with oligodendrocytes. So what are oligodendrocytes? So what are these cells? So we know that like in the skeletal muscle, the neurons are mostly they are having myelinated. They are myelinated with wrapping of squamous cells. So this myelination of the nerve in the skeletal cell, in the peripheral nerves, that makes the nerve impulse to travel very, very fast. We call that as a saltatory conduction. So in comparison to the brain, there is not proper squamous cells wrapping here, but we know that in brain also the nerve impulse need to travel faster. The information need to travel faster so that we can have a very good and stabilized reflex. So this sort of wrapping up, sort of squamous cells or sort of myelination is provided by oligodendrocytes. So let's say this is one nerve with a cell body, and there is an axon here, okay, and there is the dendrites, and there is another nerve here with a terminal bulb. Sorry, I make it big, quite big. Okay, there is a terminal bulb, and there is the dendrites here. So what is the role of oligodendrocytes? They are present between the neurons. They are present between the neurons, and what they are doing here, they are giving out some processes. So which are giving out some processes like this. So what they are giving out processes like it's same like this structure. They are also giving some wrapping up myelination. They are also myelinating the nerve here. So who is myelinating the nerve in the CNS? It is the oligodendrocytes. Okay. So one oligodendrocytes, it can wrap up even more than one. It can also come this. It can give some long process here and can also myelinate this part. Okay. So this is the role of oligodendrocytes. So what they are doing, they are making the impulse travel very, very fast. How? They are providing myelination inside the brain so that the information can travel very fast to the another neuron, from one neuron to the another neuron. Means they are helping in the faster information relay. Okay. So these are oligodendrocytes. Alright. So what does oligodendrocytes do? They help in the nerve impulse traveling faster by wrapping up the cells, okay, by doing myelination. Next is astrocytes. So astrocytes, they are not exactly like oligodendrocytes. So what they are doing, so astrocytes, okay, they have long processes. They have long processes. Astrocytes have long process, and this long process also has a end plate. It's called end plate, as a foot plate. Okay, it's a foot plate. It's a end plate. It's an end plate. It has a structure looks like this. It has an end plate like system. So what they are doing here, they are also giving structural support. So they are giving structural support. They are giving structural support to the neighboring neurons. They are giving structural support by this end plate. So let me make it quite big. So they are supporting the neurons. They are supporting. Like for example, when you see a newly constructed building, outside the newly constructed building, they make lot of frameworks so that they can climb and reach the building and can do lot of works. So that is called scaffold. Okay. So exactly like that, the astrocytes, what they are doing here, they are providing scaffolding. Scaffolding. They are providing scaffolding. That is, they are providing structural support to the neurons. They are providing structural support to the neurons. So this is one more neuron here. Okay. And they are providing structural support. They are providing structural support. Okay. So, and they stabilize the brain. They stabilize the neurons inside by providing structural support. That is called scaffold. So you see the scaffolds normally outside the newly constructed buildings. When you see the construction workers, they make a lot of framework outside the building, and they climb through the frameworks and do all the things. So similar way, they make a function of scaffold. So what is one function of astrocytes is scaffold. Another thing is that they can produce glial scar. Glial scar. So what is glial scar? So let's say this is an astrocytes. So normally astrocytes, if you see in the microscope, they look like star. Okay, they look like star because they have lots of processes here. They have lots of processes here. Okay, so they look like star in the microscope. So from where the name came from, astrocytes. So this glial, what they do, suppose there is an injury in the brain. Suppose there is a brain in the brain, there is some injury. Okay, there is some blood clot, infection, whatever happening. So what happens to the nearby? These astrocytes, they will then produce lots of processes. These processes will be thick. They will process then lot of. Okay, so they will process thick processes. This thick process with end plates. So what they are doing here, they are containing the infection, they are containing the inflammation, or they are surrounding the everything by their end plates. Okay. So that, and ultimately what will happen? Here there will be formation of a scar. So they have made a scar, and it's not infection or inflammation. They have contained it, not allowing to go outside of the region by producing a glial scar. Third thing, they also go do homeostasis. Okay, homeostasis is the maintenance of internal environment. So they regulates the potassium channels, basically potassium channels. They basically regulate the potassium channels and do homeostasis. And also these astrocytes, they can conserve inside some amount of glycogen. And when the brain needs some energy, they can convert this glycogen stored into lactate and giving a source of energy. So in the homeostasis, they control the potassium movements by controlling the potassium channels. Also they produce lactate, which is a source of energy. And number 4 is they can clear synapses. Okay, I wipe out these things. Number 4, they can clear synapse. synapses. They can clear the synapses. Now what is synapses? Synapses is the junction. Okay, synapse is the junction. Like this is a neuron with a cell body and terminal bulb. So here it will connect with another dendrite. Okay, it will connect with the other dendrite. So where it is? So it will connect with the other dendrite of a other neuron. So here the dendrites. So one neuron, when it communicates with the other, the junction between two neurons. So it is connected with the other neuron. Okay. So this junction is called synapse. So now what these astrocytes do? They try to contain the synapse, means try to protect the synapse. How? They are producing this with the end plate. So one astrocyte from this side, one astrocyte from this side. So they are trying to protect. In other way, they are making the whole this synapse, which you know, this synapse is a very important point where there is a relay of information and there is a relay of neurotransmitters. So this point is this junction, they secure the junction, they secure the perimeter from outside, so that the movement of the neurotransmitters, they stay inside, they don't go anywhere else. Okay, so that is also one thing that is called clear the synapse. They protect the synapse. Okay. Number 5 is very, very important. Okay, that is they form the blood-brain barrier. They form blood-brain barrier. Now, who can tell me what is blood-brain barrier? Blood-brain barrier. Who can tell me this? Who can tell me what is blood-brain barrier? Okay, let me ask. Shristi Nand. Can you tell me what is the blood-brain barrier? What is your idea about blood-brain barrier? Yes, Shristi Nand. She's not in the scene. Okay, Ferdinand. Why nobody responds here? Ferdinand?
Sir.
I'm asking the question to Ferdinand. Wait, not now. Not you. Wait. Srishti, wait. I'm asking Ferdinand now. Ferdinand? Okay. So Munil Nadan? Yeah. What do you understand by blood-brain barrier?
Semi-permeable border.
Huh?
It's a semi-permeable border of endothelial cells that prevents the solute in the circulating blood from entering into the extracellular fluid.
Wait, what you started, this is a semi-permeable membrane, right? You started,
Yes,
yeah, this is not a semi-permeable membrane, dear. This is not a semi-permeable. The starting is wrong. So blood-brain barrier, there is no semi-permeable membrane. Blood-brain barrier is formed by tight junctions, okay? So let's, okay, I'll tell you about blood-brain barrier. Okay, you see this, you see here. You see here, in this sample, the blood is, when the blood comes, around 15 to 20% of cardiac output goes to the brain through the carotid artery. Okay, so blood goes to the brain. Blood is moving to the brain. So means whatever is in the brain, in these arteries, so things will move from the arteries to the brain. Things can move from arteries to the brain. Things can move from here. There will be sodium, potassium. It can go outside, and it will reach the brain, right? As usual in the other tissues, but it don't happens like that in other tissues. In other tissues, in any other organs, when the blood moves, there are a lot of exchange between these extras, between these tissues and the surrounding tissues and the arteries. There are a lot of exchange, but in brain it don't exchange because the blood vessels that are flowing here, they are having in a tight junctions. Okay, so there is a blood-brain barrier, so that most of the things cannot pass through it. Only few selective things can pass through it. Okay, so this blood-brain barrier, when it is it is this blood-brain barrier which is present in the brain, it provides a foolproof security, so that anything, suppose many of the drugs, if you inject the drug in the artery, it's not necessary that this drug will go to the brain, because it is not allowed to cross the wall of the blood vessel. It is not allowed to cross the wall of the capillaries. Okay, so the blood-brain barrier, they provide security. They protects our brain from rest of the changes that takes place in the brain. Okay, it protect us from the bacteria or most of the drugs that are present in the brain blood. They can run in the blood, but they cannot go out of the blood and reach the brain tissues. Okay, so that is the blood-brain barrier. So okay, let me explain in the board here. Okay, so we know these arteries, when these arteries go, they will divide into arterioles, and then arterioles will divide into smaller branches, which are called capillaries. Okay, so let me draw a capillary. This is capillary in the normal tissue. This is outside the brain, in the outside of the brain, in the tissues, and one in the brain. So this is one capillary, and this is another capillary. Okay. So in tissue, we know there is endothelial cells. These capillaries are lined by endothelial cells. They are lined by endothelial cells. So if you watch clearly, there is a clear gap. There is a gap in most of the capillaries found in our rest of the body. There is a gap between two endothelial cells. Okay, there is a gap. Okay. Apart from that, in some other organs, there is also the endothelial cells also have some windows through which also many electrolytes can pass through. So when some capillaries have windows on the endothelial cells, we call them as fenestrated capillaries. Fenestrated capillaries. And some capillaries are neck to neck, but still there are some gap. We call them as continuous capillaries. These are continuous capillaries. And in some capillaries, like in kidney, okay, it's not well developed. The capillaries, the endothelial cells are like this. There's a huge amount of gap, where a lot of absorption is required, so a lot of things can pass through. Okay, so these are mostly found in kidneys. They are called sinusoidal capillaries. Okay, found in liver and spleen mostly. So sinusoidal capillaries, fenestrated capillaries, continuous capillaries. There are three different types of capillaries. Okay, there is some endothelial cells are neck to neck, continuous. Some have windows, fenestrated, and some are like this, sinusoidal capillaries, mostly found in liver and spleen. So there is not only three types of capillary, there is another fourth type of capillary, which is a special kind of capillary. It needs some special attention, which are found in brain. Okay, so what happens in brain? The capillaries. So let's draw the capillary. So this is the capillary. So this is the normal continuous capillary. Let's draw a normal continuous capillary. Okay, which has endothelial cell lining. Okay. So this is a normal continuous capillary. So now what happens in the brain? In the brain, apart from these capillaries are forming tight to tight, neck to neck. All the capillary endothelial cells are lined neck to neck. Okay, they are lined neck to neck, and there is no gap between through which electrolytes can pass through it. Okay, but still many of the electrolytes can still pass through it. So to inhibit that, there are some stitch. It has been stitched together. It has been stitched. Like you are making a brick wall. Okay. So you are making a brick wall like this. So if you provide some extracellular matrix between here, like cement kind of thing, it will give more and more security to your wall, right? If you put some more cement matrix here. So it will provide some more security. So same thing here. In the brain, the capillaries, each of the endothelial cells, they are stitched together. Apart from their neck to neck, they are having, so when the cells, endothelial cells are lying this way, there is no gap. We call it a tight junctions. They are tight junctions. Tight junctions. So apart from the tight junctions, we are also stitching them so that there is no chance of anything, any leakage. So they are stitched. Each and every endothelial cell, they are stitched together. Who stitches is, there's some proteins called occludins and claudins. Occludins and claudins, they stitch these endothelial cells. So when they stitch these endothelial cells, so as a result, now what happens? It's not easy to go to the brain area. It's not easy to go to the brain area for any chance. Okay? So this is a blood-brain barrier. Blood cannot pass through it. Apart from that, there is a second barrier which is giving more security is by astrocytes. So we know that astrocytes has a end plate. They have an end plate, right? So let's say this is an astrocytes. They give a security here, end plate. They are providing extra security here. So they are providing an extra layer of security along with these occludins and claudins. They are providing an extra amount of security. So these astrocytes with their foot plate, they form the important part of blood-brain barrier. Okay. So do you have any question regarding that? You can ask me now if you didn't catch up. Okay. So that is about blood-brain barrier. So because of this blood-brain barrier, most of the things they cannot pass through it. Okay. Except carbon dioxide and some other things which are very, very lipid soluble. Other things that cannot pass through it. So that's how our brain is protected from infections, from drugs. So most of the drugs, they don't reach the brain. They go, yes, they flow in the blood, but they cannot go to the surrounding blood tissues. Okay. So that is the blood-brain barrier. So blood-brain barrier is formed by tight junctions. Number one is tight junctions. Okay. There is neck-to-neck cells, and there is a glue kind of thing, and it is like stitched by occludins and claudins. And number three, there are this end plate or foot plate of astrocytes. They are providing more and more tight security. So that is about astrocytes. Okay, if you want to ask anything else, you can ask me now. So this is about astrocytes. Next is microglia. The third is microglia. So this is the third supporting cell. Okay, it also looks like this. No end plate here. So this is very interesting. This is the, you can say this is the protection. This is the police or the army of the brain. Okay, they are the resident macrophages. They are resident macrophages. So the microglia are the first line of defense in the brain. They are immune cells. Okay, they are the resident macrophage, and the most interesting thing is that they can be molded to any form. They have a plastic-like feeling, the plastic-like property. So whenever there is any bacteria or any inflammatory particle, they immediately become round. They can mold, and then they can, suppose there is a bacteria, they can swallow it like phagocytosis. They can swallow the bacteria, and they can break into pieces. Then they can break in this. Same thing, they will degrade it, they will break into pieces, which are called debris. Then they can also take this debris and present it outside. To whom? To lymphocyte. So the cell which present the antigens to the lymphocyte, we call as antigen presenting cells, APC. Antigen presenting cells. For the starting of the next part of the immunity. When then the cellular immunity, this whole inflammation process starts. So astrocytes, they are resident macrophages. They can mold themselves, they can change their form very quickly, they can swallow the bacteria like macrophagocytosis, they can chop it like and making the bacteria into debris, and they can behave also as antigen presenting cells. They can present this antigen breaking part, this debris, to the lymphocytes, and there the inflammation process starts, the cellular immunity process starts. Okay, so that is a microglia actually. And apart from that, they maintain the potassium channels. Okay, we'll discuss about this role of potassium channels later on. And they are very sensitive. They are extremely sensitive. Now we know in the blood there is blood-brain barrier, right? Blood-brain barrier. We have a tight junctions here. Okay, and there is occludins and claudins. So bacteria usually cannot pass through it. But if the bacteria, even then, it can escape. If it escapes, then what is the next step of immune level? Is that it will be attacked by microglia. It will be attacked by microglia. It is very sensitive. It can notice the bacteria, and then it can become round, it can swallow it, and it can chop it, and it can make it into debris, and it can also behave as an antigen presenting cell. So they are like the resident macrophage. But the only difference between the macrophage in the other part of the body, this macrophage in the other part of the body, apart from the brain, they cannot change their shape. But here they quickly change their shape, and they can do all the functions of macrophages. And they are very fast and very sensitive, remember that. And next is what is called ependymal cells. So ependymal cells, they line the CSF. They protect the CSF. So where the CSF is flowing from the third ventricle to the fourth ventricle, so all over these ependymal cells are protecting the CSF, so that CSF don't leak outside. Okay, so these are the 4 unique cells found inside the brain. So oligodendrocytes, number 1 is oligodendrocytes, number 2 is astrocytes, so number 3 is microglia, number 4 is ependymal cells. So these are the 4 different type of cells which are providing support to the neurons, which are helping the neurons to provide information faster, and also it has the protective functions. Alright, so this is about the neuroglia, or the 4 types of supporting cells. Okay, so other than that, now we come to the brain. So in the brain we will get 2 things here. One is sulci, another is gyri, or sulcus or gyrus. Okay, so in the brain you will see sulcus and gyrus. So you see there are some lots of folded here. Okay, if you open one fold from here, you can make it long, okay, like a strip. So each of these they are folded so that there can be lot of surface area. Okay, and each of this fold, so there is a swelling here. This is all bulge because of the folding. They are all bulging. Okay, so these are called gyrus. They are called gyrus, or in plural gyri. Okay, this foldings, this bulging is called gyrus. So it's like if you write any information in a paper and then you fold it many times. Okay, you fold it many times, and you fold it and you fold it and fold it, so it makes one gyri. So inside gyri there are a lot of information. Okay, many informations are written here. Okay, so each of these are making and if you open the gyri, it will make a long in a meters. So there are lots of information in the gyri. So this outward bulging is called gyri, and the inside between the gyri you can see there is a groove. So these are called sulcus or sulci. Okay, these grooves are sulcus. Okay, and this bulging, okay, they are called gyrus. Alright. So the brain is having 4 lobes. So from the front, we call it, this front is frontal lobe. So when you say frontal lobe, the back, so here is like a canal here. Okay, this is called as central sulcus. This is the main sulcus, the central, which is differentiating the brain from the parietal lobe. So this is the frontal from the parietal lobe. So there is a frontal lobe here, there is a parietal lobe here. So on downside there is a here temporal lobe here, and on the back it is the occipital lobe here. Okay. So this part is the frontal lobe towards the front. Okay. If I stand sideways, this is frontal lobe, and the frontal lobe is separated by the parietal lobe by a central sulcus. There is a canal here. Okay, central sulcus. Okay, there is a central sulcus. Okay, so frontal lobe is separated by the parietal lobe by central sulcus. So frontal lobe is also separated by a temporal lobe. This is the temporal lobe by a sylvian fissure. Okay, this is lateral sulcus. It's present on lateral. So lateral sulcus, central sulcus. So frontal lobe is guarded on the back. This is central sulcus. This central sulcus, and separated from the temporal lobe, this is lateral sulcus. Okay, this is lateral sulcus. So this is frontal lobe, parietal lobe, temporal lobe, and the back is occipital lobe. So these are two cerebral hemispheres. You see, these are two cerebral hemispheres. These are two cerebral hemispheres. So we know that our left side of the body is controlled by right side of the brain, right cerebral hemisphere, and our right cerebral hemisphere, right side of the movement are controlled by left side of the brain, left cerebral hemisphere. So you can ask me that is the brains working independently? Then I will say no, the brains are not working independently. Though the left side is controlling the right half of the body, but they are constantly communicating with the right side of the brain. Okay, so look, these are two independent hemispheres. So, and they are continuously connected by a set of nerve fibers. There are almost 100 million neurons here. 100 million neurons. This is called corpus callosum. Okay. Corpus callosum. So this is the corpus callosum. So corpus callosum is a C-shaped structure, and it has got 100 million neurons. So these 100 millions, they connect with the one cerebral hemisphere to the other cerebral hemisphere. They connect. They're connecting with them. They're connecting means they are communicating. One hemisphere can communicate with the other hemisphere through this corpus callosum. So that's why the two hemispheres, they work together. Now what happens? Let me tell you this thing. Now what happens when there is epilepsy, when there is seizure? Okay. So when there is a seizure in one part of the brain, when seizure is because of some hyperactive foci, okay, there is some electric generation in one part of the brain, and then it is causing seizure, epilepsy. So this seizure, if it is developed in this part of the brain, yes, it can develop right side of the body also. But what will happen? It can also stimulate the right side of the right hemisphere also, because it is well connected. So when the treatment of the grand mal, when the grand epilepsy, okay, we will not discuss epilepsy, but so one of the type of treatment of epilepsy is that when it is not treated and when there is severe epilepsy, then this corpus callosum is removed, which we call corpus callosotomy. This corpus callosum is removed. So when the corpus callosotomy is removed, then when the epilepsy generates in this lobe, it cannot go to the other lobe, and it's protecting the brain and the rest of the part of the body. So that is also done. Now what will happen? Just imagine that you remove this corpus callosotomy. So during that, you had a patient of seizure, and there's a lot of problems, and finally the corpus callosum has been removed. So we call it corpus callosotomy. Corpus callosotomy. You did that operation and remove this corpus callosotomy. So what will happen now? This part of the one brain cannot communicate with the other part of the brain. So what will happen? Imagine. So means, let's say this is a picture where you can see, so many things are there. There are some objects here. Okay, there are many things, objects in the one picture. So if I ask you to draw and copy this picture, okay, so what will happen? Suppose, so what will happen here? you'll not be able to draw half of the picture, because when you see now let's see this picture. This is left side, this is right side. So left side of the picture goes to it cross. So when it goes to the brain, to the occipital lobe, it cross, the fibers cross. It goes to the opposite side of the brain. So right side of the fibers, we take this image, it goes to the left side of the occipital lobe, left side of the brain. So if one brain is not communicating with the other brain, so what will happen? You will not be able to read left side or right side, depending upon which side of the brain you are getting. Okay. So if I remove the left callosotomy, so this is my this is the right hemisphere. If I remove the right corpus callosum, so right brain, it take the signal from left side. Okay. So if you remove the right callosotomy, now though you receive it, but you cannot convey the image to the left side. So hand-eye coordination, hand-eye like feeding, hand-eye coordination, when you are observing from the lateral side, is processing from the right side of the brain, right side image are processing from the left side of the brain, everything got disturbed. Most of the centers, the learning centers, this centers, that center, most of the centers in the brain, they are always bilateral. There's one in the left, one in the right. But our language system, the way we learn the language, that is only present in the left side of the brain. That is only present in the left side of the brain. Remember that. Okay, the language system, center, what we call as Wernicke's area. Wernicke's area. So Wernicke's area is present only over the left side of the brain. So if the corpus callosum is removed, then what will happen? There will be miscommunication between the two lobes, and they cannot integrate, they cannot do the work properly. Like for example, you are riding a bicycle. So when you ride the bicycle, we use both the legs, pedaling. Means left leg is governed by right side of the brain, and right leg is governed by left side of the brain. So there is a continuous communication between our two hemispheres. So if corpus callosum is removed, left side can be goes to right, but right information cannot go to left, and there will be proper discoordination of movements. Like in dyslexia, most of the features, like this is, so there's a language disorders, learning, hand-eye coordination, like feeding. These things are not developed properly when there's a corpus callosum is not developed properly, or it is surgically removed. Sometimes corpus callosum is not properly developed in some children. Then also they have a lot of feeding problems. They cannot feed themselves because their hand-eye coordination is needed. They cannot ride the bicycles in those children. So sometimes corpus callosum is not properly developed, or sometimes it is surgically removed. Then it can create a lot of problems. Okay, we'll come to each of the lobe in detail in the Wernicke's area speech. We'll come to that in detail. Today is just an overview. Okay, so we are talking about frontal lobe. So frontal lobe is the largest lobe of the body. It is the largest lobe. Okay. So frontal lobe is the largest lobe, and it is demarcated by central sulcus on the back and lateral sulcus. So lateral sulcus differentiates the frontal lobe from the temporal lobe, and the central sulcus demarcates the frontal lobe from the parietal lobe. Now where there is the central sulcus, you can see here, wait. So this is the central sulcus. So this area before the central sulcus, this is called precentral area. This whole blue area is precentral area because it is just before the central sulcus. Okay, this is called precentral area. So this precentral area, both sides, they are concerned with all the motor movements. Okay, so if you are moving your left hand, then it is done by the right precentral area. So if you are moving your right hand, it is done by the left precentral area. So this is the area for the motor functions. This is the area for motor functions. So what is this area? This is called precentral area in the frontal lobe, just before the central sulcus. Okay, so what else in frontal lobe? So frontal lobe is needed for all the executive movements. Like you plan for future, that is by your frontal lobe. Like inhibition, social adjustment, okay, that is done by the frontal lobe. the emotions, that is done by the frontal lobe. Look, for example, if you are going to a funeral, you need to show some emotion, right? You cannot go to a funeral and laugh around. That is because totally opposite to the situation. But if someone have a frontal lobe disorder, they don't act according to the situation. They don't act according to the situation, and they will behave in an opposite manner. Okay, in a sad situation they will laugh, in a laugh situation they will cry. So this is all because of frontal lobe. It is the lobe of emotions, lobe of judgment, lobe of execution of functions. When you are planning for future and how you execute it, the attention span is provided by the frontal lobe. Okay, and plus it is also responsible for inhibition, social inhibitions. Like people are shy because of frontal lobe. So many of the times you see there are some psychiatric problem in the patients who walks nude. Okay, he undress in the front of public, he remove clothes of theirs in the front of public. Why? What? When will it happen? When there is a frontal lobe disorders, because of social inhibitions. We have social inhibitions. Okay, we don't do everything in public. So that social inhibition is lost if there is a frontal lobe disorders. The attention span is lost if the frontal lobe disorders. And plus this frontal lobe is responsible for short-term memory. So short-term memory. So work type memory, short-term memory, you remember in the numbers, all the short-term memories are by the frontal lobe. Alright? So other than that, frontal lobe is responsible for motor functions. Remember that. The precentral area is the area of motor. We'll discuss that more when we do the motor functions. Next is parietal lobe. So where is the parietal lobe? The next, behind this central sulcus is the parietal lobe. This is the parietal lobe. So what does parietal lobe do? So remember, so this is the central sulcus, and the front part is precentral. Obviously the back part behind the central sulcus is postcentral area. So sorry. So this postcentral area is responsible for sensory functions. So whatever the sense you feel, touch, pain, pressure, temperature. Suppose someone is touching, then you can feel the temperature, you can feel the touch. Where you are feeling the touch? Because this, whenever someone pricks or someone touch, then from here the nerve will go and it will terminate where? It will terminate in the postcentral area. It will terminate in the postcentral area. So this postcentral area is made for sensations. So any type of sensation, touch, pain, pressure, vibration, all these sensations are related to the postcentral area. So if there is a clot or there is an injury and affected the postcentral area, then the person will lose the sensation. If touching the patient hand, he will not feel it. Okay, he is paralyzed and he is not feeling the sensations. Okay, he is not feeling it. If there is any cut or if there is any, you're touching it, he is not feeling. Why? Because it affected the postcentral area. Now sometimes there is a paralysis when patient cannot move his hand or move his legs, move his fingers. Why? Because the problem is in the precentral area. So when there is the precentral area, that is motor, postcentral back side of the central area, that is for sensory. Remember that. So other than that, what else parietal lobe do? They sensory, yes. The frontal lobe is for motor. It is all the actions like speaking, movement of the mouth, all these are done by the frontal lobe. All the sense, your feeling is done by your parietal lobe. Okay, that is by the parietal lobe. And then we come to the temporal lobe. So temporal lobe is separated from the frontal lobe by this lateral sulcus. So temporal lobe is very, very important for in terms of memory. All the long-term memory is in the temporal lobe. Plus the language, okay, learnings, most of the things like new skills, okay, development of long-term memories, execution, all those things are in the temporal lobe. The main seat of memory is hippocampus. So hippocampus is continuously talking to our temporal lobe and making the short-term memory into long-term memory. Okay, so when we sleep and close our eyes, then that time the hippocampus along with the temporal lobe, they remove all the unwanted functions and they keep the important memories and put them in from short-term they make it into long-term so that you never forget. Okay, so that is a long-term memory. Then on the back This is the occipital lobe. On the back, this is occipital lobe. So occipital lobe is needed for our vision. It's needed for our vision. Okay. So when you see any object, whatever image you are seeing, our eyes are just transmitting that. Our eyes is like a camera. It is capturing the image. So through the nerves, it will come in here. Through the nerves, it is going to the occipital lobe. But we'll discuss in detail when you study the vision. But from the left eye, most fibers are going to the right occipital lobe. From the right eye, most fibers are going to the left occipital lobe. So there is a part in the midbrain, above the midbrain, where these fibers are crossing each left side of the fibers are crossing, and right fibers of the crossing to the left side of the fibers are crossing to the right side, and that point is called optic chiasma. Okay, we'll discuss that in detail. Today just overview. So occipital lobe is needed for our vision. Now let me give you an example. Now suppose somebody got hurt in occipital lobe. So what you will lose? You will lose vision. Okay. Now just for example, it comes as a reflex. When you can't see anything in distance, anything clear in a distance, you're trying to see, focus, and then you mostly scratch your back head. What you are? That's a reflex. There is no scientific advantage of that, but we are trying to stimulate the occipital lobe actually, because you are trying to focus. Who is that person? I don't know him or her. I'm trying to stimulate my occipital lobe. So because vision is needed. So when a patient comes and I am having deep headache, where? In my back of the head. Then we should say, go check your eyes. Your glass, check your glasses, your power, refractive power, eye power must have changed. Check it. Okay. So that comes with this, because occipital lobe is concerned with vision. So when there is a problem with vision, when our eyes are not adjusted properly, when our glasses are not adjusted properly, we'll feel a deep headache. Where? In the occipital lobe. Because they are concerned with our vision, the color vision, black and white vision, all those vision, formation of image, it forms in the occipital lobe. Now you are in a deep trouble, you are having a lot of function, you are financial problems. So you see, when you are not understanding, you are confusing, you are always scratching frontal, because frontal lobe is needed for the judgment, for the decision, for the all those understandings. Temporal lobe is, it is the intellectual abilities, music skills, other skills, memories. When you are trying to recall some long memory, I forgot the face, what happened that day, you are scratching your temporal lobe. You'll see it happens automatically. Okay, but I'm just trying to make you understand. There is no scientific on that. Don't write that thing on the paper also. I'm just trying to make you understand. It comes automatically when you're trying to remember some long incidents. What happened? Somebody's trying to recall, giving yourself. You didn't remember what happened 10 years back. 10 years back is because you are automatically you're stimulating your temporal lobe. It's giving you long-term memories here. When you're confused, tensions, all financial problems, confused, what to take, what should I do? What should I do? You are always scratching your frontal lobe. You are not understanding anything, then go for parietal. You are not feeling anything, not understanding, then you can go for parietal lobe, which is needed for sense, like 2-point discrimination. When someone touch with 2 point, with a forceps or scale with touch 2, then with parietal lobe we can know that he has touched 2 point on my body. If the parietal lobe don't work, we cannot differentiate between 2 point. This is 1 point, second point. If we touch simultaneously, I'll say it 1 point. If I close my eyes, I'll say it 1 point. But if my parietal lobe is normal, and even if you touch like this, I'll say you have touched 2 point. Okay, that 2 point discrimination, that is my parietal lobe. Okay, so frontal lobe is the action, all those workings, lifting of hand, lifting of leg, walking, okay, talking, swallowing, all the movements of our body. Eye movements, mouth movements, speaking, all these are frontal lobe. Memory, short-term memory is by frontal lobe. Long-term memory, temporal lobe. You're trying to remember what happened yesterday, or just 10 minutes back. What happened yesterday? 10 minutes back, she was saying something, I forgot. Then use your frontal lobe. Don't scratch your temporal lobe, it will not process. Okay? And you can't see properly, stimulate your occipital lobe. So that's how we differentiate. If a patient is paralyzed and patient is not able to judge, is not losing all the inhibitions, okay, losing all the social inhibitions, no attention span, many of the things, then we focus on frontal lobe. There must be some frontal lobe disorders. And if the patient cannot feel any senses, then we focus on parietal lobe. And no memory, okay, then temporal lobe, and vision is lost or vision has been compromised, we focus on occipital lobe. So these are the 4 lobes of the brain, okay, which are connected between by corpus callosum. Okay, so they are connected by corpus callosum. So you see here, this is corpus callosum here, and now what is this? See, so with this thing you can hold the brain. You can hold the brain with this thing. So that's why it is called brain stem. This is called brain stem. Okay, so this part is the midbrain. So it is in the middle part of the brain. So midbrain, then pons, medulla, and you can see there is cauliflower-like structures. Cauliflower-like structure. This is below the cerebral cortex, so we call it the cerebellum.